In this video, we will discuss about the effect of FASL gene knockout on the CAR T cells on its anti-tumor potency. Since we have seen that when the FASL gene is been knocked down, it will increase the CAR T cells efficiencies by reducing the apoptosis that is triggered by this FASL gene activations when the CAR T cells gets activated. They wanted to see whether this knocking out of FASLG will reduce its anti-tumor potency against the case where FASL gene is present in the CAR T cells. So for this they used 3 different cancer cell lines against which they checked its effect. And those cell lines are Raji cells, null M6 and JVM2. These are you can consider as B cancer cell lines. And this is the plan that they used. Is they knock down FASL gene in the T cells, and in case and simultaneously they transduce these T cells with the 1928 or CD19 extracellular domain and CD28 in the cellular domains part. And in order to maintain the homogeneity, what they did is they transduced the cells after enriching the cells with CD8 plus T cells populations. And in panel B shows the data from the literatures about the FASL presence in the cancer cells. That is null M6 has this median level, and the Raji and JVM2 is in the upper side of this plot that we can see it here. And this FASL, they also check the FASL expressions related to the transcripts that are found in the literatures, and they can see that null M6 lies similarly in the like parallel or proportional range. That is, with increase in the FASL transcript, they see FASL relative expressions also increase in the null M6. Now for the null M6, they check the FASL expressions when they are transduced with when they are transduced, or no, they are not null. B cells are not transduced. They check the FAS expressions in the null M6, and we can see that null M6 FAS expressions are high compared to its isotype case. And thereafter they check the effect of the effect on the null M6 when the CAR T cells are when they are treated with co-cultured with CAR T cells at different ratio. It is effector to target ratio 1:2, 1:4, 1:8, 1:16. And for the control case, they can see that when both of them culture separately, there is no significant change in the or there is a no significant change in the increase in the M-cherry signals. But when they co-cultured together, we can see a decrease in the M-cherry signals, which is the indications for the B cells or the NLM6 cells. When they are lysed, its M-cherry signals goes down, and we can see there is no significant difference when they are cultured at 1:2, and at 1:4 they also see no significant difference, and for 1:8 also they do not see any significant difference. But when 1:6, that is when the target cells are present in very low amount, they can see that for the knockout case they see more cell death compared to the wild type case. So this indicating that this results indicates that CAR T cells FASLG knockout on CAR T cells does not reduce the anti-tumor potency, as the knockout cells has more cell death even at when the effector to target ratios are very low. Thereafter, they check the similar results for the Raji cells. In the Raji cells, they can see that FAS expressions are really high, and when they check the effector to target ratio, in that case also they do not see any significant difference for 1:2 and 1:4, but for like 1:8 they see a significant difference. But in that case also, for the knockout, they see a high cell death or low M-cherry signals compared to the case where CAR T cells are being transduced with FASLG, and for 1:16 they do not see any significant difference. And when they check it for JVM2 cancer cells, And this here also they see the similar results. Or we can say they do not see any significant changes in 1:4, 1:8, 1:16, but they see significant changes around 1:2. In that case also they see that for the knockout the cell death are higher, or in the case where FSL gene is been knocked down in CAR T cells that are transduced to the CD8 plus T cells. So in together they finally conclude that the FSL gene knockout does not reduce or does not cause the loss in the antitumor potency of the CAR T cells against the B cancer cells, as they had tested for the different kind of B cancer cells. So that's all for this video. I hope you understood their hypothesis and how they check their hypothesis and their final conclusions. If you need to understand more about their experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.